The human visual system begins with the eye, which is nature's camera. The eye contains many of the same parts as a camera. There's a series of lenses that focus light, the cornea, and the lens of the eye. Light is collected through the lens, which changes its shape to focus the light by the action of specialized muscle cells called ciliary muscles. Just as a camera records light with a sensor, light is recorded in the retina by a set of specialized light-sensitive cells called photoreceptors that convert light energy into neuronal activity. And just as a camera has electronics inside that process the image and transmit it down a cable, the retina does a fair bit of fancy image processing and is responsible for transmitting the image signal down the optic nerve. The eyeball itself is able to pivot around thanks to oculomotor muscles, not unlike the panhead on a tripod. And there's a fairly advanced circuitry in the brain for automatically moving the eye to stabilize the image, not unlike the vibration reduction feature of many lenses. The eye can also adjust to differences in illumination by controlling the pupil diameter, which is equivalent to adjusting the aperture on a camera, for those of you who are photography buffs. And as an added bonus, the eye is able to clean itself by producing tears and blinking, and it can even repair itself in the event of modest damage to its optics. By basically any measure, the eye is a remarkably sophisticated camera. The human eye has as many as 100 million photoreceptors, but they're distributed in an extremely uneven way across the retina. In particular, there are way more tightly packed photoreceptors at the center of your gaze in a region called the fovea. For this reason, you have very high-resolution vision only in a very small region of visual space, roughly the width of your thumb at arm's length, at the center of your gaze, and resolution falls off dramatically after that. You only think you see the world around you in high, crisp resolution because your eye is ceaselessly moving about 3 times a second on average. If you try really hard and keep your eyes fixed on this cross, then you can get a sense of how blurry your vision is elsewhere on your retina. Only 1 million axons or so carry information away from the retina, so in some sense, the retina is only a 1 megapixel camera, fairly unimpressive by the standards of camera specs. However, because your eyes move around and sample the visual scene, and because a lot of fancy processing happens in the retina, which we'll get to in a bit, that 1 megapixel spec is a bit misleading. It should be noted that not all animals have retinas arranged like ours. While primates do, most mammals don't have a high-resolution fovea, and some have other distributions of higher and lower density for photoreceptors. For instance, some herd animals have a horizontal streak of photoreceptor density, possibly to help them keep an eye on threats approaching from the horizon. And of course, invertebrates, like insects, have completely different architectures for their eyes, including many lenses arrayed across the eye. Vision in invertebrates is fascinating in its own right, but for the sake of time, we're going to focus on vertebrate brains in this lesson. So how does the vertebrate retina work? Anatomically, the retina has at least 10 distinct layers, which can be simplified into 3 main functional stages with 2 synaptic layers: photoreception, internal transmission, and output into the deeper brain. Interestingly, these layers are arranged in an inside-out fashion. The processing of light occurs first in the deeper layers, farthest away from where the light enters. As mentioned already, light detection occurs in photoreceptors, and we'll learn the details of this process later. Once detected, the light signal is transmitted synaptically and processed by a series of interneurons, including bipolar cells, and further passed on synaptically to the retinal ganglion cells, which act as feature detectors. At each synaptic stage, there are laterally connecting cells, called horizontal and amacrine cells, that allow synchronization and some integration of the signal input. Interestingly, while there are only a handful of different types of photoreceptors, there are more than 50 different types of interneurons and more than 30 different types of retinal ganglion cells that have been identified. This allows for a very detailed differentiation and integration of the visual input. Axons of the retinal ganglion cells are then bundled into the optic nerve, which distributes visual information in the form of parallel action potentials that code for each visual feature detected. These features are then sent on to several deep brain regions that are highly specialized for the visual system, but more on that later.